folks. Welcome back to week 7A, integumentary system. This is the third and final lecture in this series. And in this one, we're going to learn about the dermis, sensory receptors, and structures related to the hair. So let's start first with the dermis. So we're completely done with the epidermis, and we are going to be going deep to the dermis. So in the picture on the bottom right, you can see that the interface between the dermis and the epidermis is not a straight line, but it's more of an egg crate type of structure going on. And in the epidermis, we are going to divide it into two areas. So let's look first at the photomicrograph on the left. The darker purple cells at the top, that is the epidermis. So my pointer is going along the border of the epidermis, okay? And if you look just deep to the epidermis, the more superficial part of the dermis you can see is like as if you had some pink chalk and you kind of smudged it through. And then if you go deeper to that, you can see like some big, thick bands of stuff going through. So I think you can clearly tell that there is a difference between the more superficial part of the dermis and the deeper part of the dermis. So the more superficial part of the dermis is lighter pink smudgy area. This is known as the papillary dermis, which is about the top 20% of it. And in the papillary dermis, we have these upward projections that you can clearly see in the bottom right cartoon drawing. And those projections, each one is called a dermal papilla. So this is the singular form, and up here in the text I wrote the plural form. So if I'm only pointing to one, please don't put that final E on it, just give me the singular form. And the papillary dermis is made out of areolar connective tissue. And so that's why it has this more homogeneous appearance. It's going to have lots and lots and lots of capillaries, which are needed because the nutrition needs to come from those capillaries and diffuse upward into the dermis because the dermis is avascular. There will also be some sensory receptors and some nerves as well in the papillary dermis. The deeper part of the dermis is called the reticular dermis. So the reticular dermis is more than half of the dermis, almost 3/4 of the dermis or more. And it's not areolar connective tissue, it's dense connective tissue. You can see these big bands of collagen, but I think you can also notice that they're not all aligned in the same direction. They're going a whole bunch of different directions. So you can pull your skin like this, pull your skin like this, pull your skin like that. And so this is dense, irregular connective tissue. We're going to have hair structures going to the reticular dermis. We're going to have sensory receptors. We're going to have nerves. We're going to have glands going down in here. We still have blood vessels, not quite as prominent as in the papillary dermis, but still plenty of blood vessels. So let's look at these receptors. So there are 5 different receptors I'm going to be talking about. And when I'm done, come back and look at this picture because it's an unlabeled picture and figure out which one is which. Now, some of these do have multiple names. I do tend to stick with the first name I've listed, which I think are the names that McKinley is given. But if you're repeating the course from somebody else and they use these other words, you can go with those. If you haven't, I really hesitate doing the tactile thing because if you notice, 2 of them are called tactiles. The only difference is one's a disc versus a corpuscle. So if you can just stick with these other words, it would be easier, I think, for you to learn. So starting off first, we have free nerve endings. What does this mean? This just means these are the sensory dendrites of the neuron, okay? And they can be located in the dermis, as seen in the picture on the left. So the arrow in the picture on the left, that would be a free nerve ending of the dermis. Whereas the picture to the right, where you see it branching in the cartoon drawing rather than a photomicrograph, those are going up into all the various layers of the epidermis. So that would be an example of a free nerve ending of the epidermis. And these can sense temperature and they also can sense pain. And they look exactly the same whether or not they are censoring hotter temperatures or cold temperatures, and they're just going to send their information. So the ones that sense cold temperatures sense at this temperature range. I put them in Celsius because I don't want you memorizing temperature ranges, okay? So cold basically is anything that is going to be colder than a normal body temperature. And warm overlaps about 10 degrees Celsius lower than a normal body temperature to about 10 degrees warmer than a normal body temperature. And so that's warm. If you notice, there is about a 10-degree overlap where both cold and warm free nerve endings will be stimulated. Okay? And that's how your body can say, oh, I'm comfortable. I'm neither warm nor cold, because you have the equal amount of sensory information coming in. So if you've ever noticed on a hot summer day, if you're down in the South, they give you sweet tea with some mint in it. And that's because menthol, if you've ever had a peppermint or a spearmint, it stimulates cold receptors. So even though you're not cold, you're faking out your body by stimulating your cool receptors. So you actually feel cooler than you otherwise would. And similarly, but exactly the opposite, there's a substance in peppers, and not your bell peppers, but in your spicy pepper, called capsaicin, and that stimulates hot receptors. So on a cold winter day, that's why we like to eat spicy foods and chilies and things like that, because we're stimulating these hot receptors. And one other really cool, interesting thing is when the temperature gets super cold, so right around freezing and a little bit above freezing, not only are we stimulating our cold receptors, but we can stimulate our pain receptors. So if you know people who live in cold environments, like for instance, I lived in Montreal for 8 years, where every February it is minus 40 degrees. You go outside, your skin hurts. Yes, it is so cold that the pain receptors are being stimulated, so it hurts, which I think is probably a good thing, because it tells you time to get your you-know-what back inside, or to dress better for the outside. So those are the free nerve endings of the dermis and the free nerve endings of the epidermis. Be sure to write that word dermis or epidermis so that you are being accurate with labeling it. The second sensory receptors are those Merkel cells, okay, are attached to the Merkel cells. So here's the Merkel cells, and we're going to call these Merkel disc. So go back to when you used to have dates, okay? And if somebody came, or when you were a kid and you were playing with your siblings or your parents, take that feather and run that feather along the surface so that only the ends of the feather are touching your skin. The only sensory receptors being stimulated would be these Merkel discs attached to the Merkel cells. So these are receptors for very light touch. That's why they're in the epidermis. The next type of sensory receptor is called a Meissner corpuscle. And to see these, you need to go look at the cartoon drawings where you have the dermal papilla, because these are located in the upward dermal papilla, not in all of them, okay, but in some of them. And they look like the cotton part of a Q-tip, okay? So they're only found in hairless skin, and we have lots of them on our fingertips, lots of them on our tongue, lots of them on our lips and our nipples and our reproductive organs, the penis and the clitoris. And what these are responsible for is something called 2-point discrimination. Now, we do have them elsewhere, but we don't have lots of them. So this is an interesting experiment. If you're bored, you can kind of do it with yourself, and that is take 2 pins. And if you were to poke yourself with 2 pins on your back, as long as you were close than about 10 centimeters apart, if you did them exactly on the same time, like if you took a staple, you wouldn't be able to tell if you were poking yourself with one of them or with 2 of them. Because we don't have very many of them on our back. Now we have more of them on our arm, so you would have to be a couple centimeters apart to not be able to tell. Okay. But on our fingertips and our lips and our tongues and those genital areas, we have so many of them that you have to be a millimeter or two apart in order not to be able to tell, because we have so many of these that you will still be able to tell that there's two and not one. Once you're more than about a millimeter apart, you can tell you're being poked with two. All right, another type of sensory receptor is called a root hair plexus. So plexus, I know it's a pink drink that some ladies buy. I don't know, it's supposed to help you with weight loss or something. Don't do it. But a plexus in anatomy is when we have different nerves whose axons, whose structures are kind of coming together and going apart. And so a root hair plexus is meant to tell you it's more than just one neuron that's being involved here, more than one nerve. Okay? The drawing on the left, it looks like it's just one going there, wrapping around part of the hair. But the picture, that's the photomicrograph, you can see that there is part of a green neuron, there's a yellow neuron, and there is a red neuron. So every single hair has more than one nerve cell coming in and giving sensation to the deep part of the hair. And what this can recognize is hair movements. So if you've ever seen those ladies, like if you're at a bar, like in high school, you saw those girls in your class who were flirting and they were flipping their hair, and you would say something to them like, quit flipping your hair, and they go, what? Yes, they can tell, because every single one of those hairs that they're flipping is giving the little message to the brain, you're moving your hair. Okay. Now, guys, before you laughed at the girls, I got a bone to pick with you, because when you were in grade school and you were sitting behind a girl and you pulled on one single hair and she go, teacher, Johnny's pulling my hair, and you go, can't tell it was one freaking hair. Yes, she could tell, because you were moving one hair that has a root hair plexus on it. Okay, so you kind of owe us the apology for that one. All right, now I think the last one I'm talking about is called the Pacinian corpuscle. And if you look at it, it kind of looks like if you took an onion and squished it down and cut it in half, where you can see all the layers to it. Because that's what it is. It's lamellar. It's got a bunch of different layers. Now, these are found very deep in the dermis. They're not going to be found by the derma papilla. They're not going to be found in the middle, but they're going to be found deep in the dermis. And these are also found in the hypodermis as well. And the reason why they're found in deep locations has to do with their function. They are for deep pressure and for vibration. So by deep pressure, I mean like when Olga is walking on your back, giving you that deep tissue massage, not that little one, like, you know, when you got the little feather going, or you're doing this little facial massage to get the blood flow going. Lamellar corpuscle, a deep massage. And they also come into play when you're doing that manly activity with the vacuum cleaner, you know, if you're pressure washing something or digging up concrete or something like that. All right. So here is a picture of an artist playing a guitar, or it might be a bass. I don't know, because I don't know my instruments that well. And so I want you to stop and think for a second as this musician is playing music, and let's just make it a bass just for continuity. What sensory receptors do you think are involved with him playing this guitar or bass? And when you're done with that, we are done with this third talk as far as the receptors are going. So go ahead and pause it now. Okay, now that you're back, I'm going to move on and talking about hair. And this is the last part of the integumentary system. And so on the right, you have a scanning electron micrograph of a hair coming out of the skin. And on the left, you have the deep part of a hair in a regular photomicrograph. So we've got a lot of hair on our head. Some of us have more than others, okay? Gentlemen, that male pattern baldness is inherited from your mom. So look and see what her siblings and her dad looks like to see if you're gonna have that male pattern baldness. Ladies, it doesn't matter. Once you hit menopause and your hormone levels are going to go down, you're going to thin out up there as well. So there are parts of the human body that don't have hair. We don't have any on our palms or our hands or our soles of our feet. We don't have any on our lips, our nipples. We can have them around our nipples, but not on the nipples. And some external genitalia has hair and some does not have hair. So what is a hair? It's dead. What you are seeing is dead. Dead cells, dead keratinocytes. So basically all you're looking at are shingles of keratin. The first thing to know about every hair in your body, it has its own private little muscle. This structure right here, which is attached to the hair on one end, and it's attached to the superficial dermis on the other end. And what happens when this muscle contracts, that makes the hair stand up, and that's what happens with goosebumps. The name of this muscle is the erector pili muscle. So looking at your hair, I talk about this in more detail in the lab lecture, but I'm just going to review it here. Everything that you can see on the outside, everything that is external is called the hair shaft. Everything that is in the skin, whether it's in the epidermis or the dermis, or whether in some places in your body goes down into the hypodermis, everything that is hidden is called the hair root. The deepest part of the hair root has a swelling. You can see it's thicker than this more superficial part of the hair root. And this is the bulb of the hair root down here. What you should notice is that the entire hair is surrounded by epithelium, epidermis. So right here is the stratum basale of the epidermis, and when it gets to the hair, it goes down and completely surrounds it and appears on the other side. So it's not just doing it in two T's from the two sides, it's doing it completely surrounding the entire hair structure. And the epidermis, the epithelium that's surrounding the entire hair root, is called the hair follicle. And down here at the base, this is where we're going to have the germ cells to be making the new cells of the hair. Going back to looking at the bulb of the hair root down here, this little red flame thing, that is actually not part of the hair. That is something that's inserted into the bulb. And what that is it has like a little arterial blood vessel bringing nutrition. It has a little venous structure draining the waste. And that entire structure is called the hair papilla. That's it for hair. So now let's talk about the glands of the skin. In the skin, we're going to have sebaceous glands, which have an oily secretion, and we're going to have sudoriferous glands, which are sweat glands. Okay. And so the first thing I need you to do is go back and think about that first lecture in this series where I talked about coiled tubes versus blobs. Okay, because that's how you're going to tell the difference between a sebaceous gland and a sudoriferous gland is by their structures. So sebaceous glands always will be attached to a hair root. And these are the glands that have that blobby appearance, okay? So they are branched, holocrine. You don't have to know what that word means because I told you I was skipping that for this semester. They produce an oily substance. That substance is known as sebum. That is where the word sebaceous comes from the sebum. So know that word sebum. And what the sebum does is it coats the outer surface of the hair as well and your skin, which softens and lubricates it and keeps it very flexible until you put your shampoo on, which strips all the sebum on, and then you have dry, gross hair, and then you put your conditioner on to try to soften it again. So you're spending money to rip this off, and then you're spending more money trying to put it back, something man-made back on. So those are your sebaceous glands. And the interesting thing about sebum is antibacterial. Your hair conditioner, not antibacterial. Okay. And gentlemen, I know I wanted you to apologize for pulling our hair in 3rd grade. And ladies, we owe gentlemen an apology for high school when puberty hit in. And ladies, you started your skincare routine and most of you managed to control your acne, whereas the guys had much worse acne than you did. And you were just thinking, well, man, why don't those dudes wash their face? It's because they were having massive amounts of testosterone, and ladies, you weren't. And testosterone is an androgen which stimulates the secretion of the sebum. So that's why their skin is so much oilier than ladies' skin. All right, so now we're going to move on to the sudoriferous glands, or the sweat glands, and there's 2 different types here. And histologically, the cells look exactly the same. So what you need to do is see where the duct is going to. So the first type of sudoriferous or sweat gland is called an eccrine gland. And these are widespread throughout your entire body. We got tons of them on our palms. We got tons of them on our face and our foreheads. And the way you can tell a eccrine sweat gland, sudoriferous gland, is because number 1, it's a coiled tube instead of a blob, so that makes it sudoriferous. And the second thing, if you follow the duct goes all the way to the surface of the skin. So over the picture in the middle of the screen on the left is the cartoon drawing showing cross sections of the ducts, and then you can see the tubular part of the duct going to the surface. And then on the right is a photomicrograph of a similar type of sweat gland where you see it going all the way up to the epidermis. You don't have to see it going all the way up to the surface like the picture on the right. If you see the duct going up to the epidermis, that means it's going to the surface. So those are the eccrine sudoriferous glands. The other type of sudoriferous gland is called an apocrine sudoriferous gland. And if you follow its duct, what you will see is its duct always dumps and surrounds the hair follicle. So hair follicles will always have a sebaceous gland and may or may not have an apocrine sudoriferous gland. So one hair can have both or can only have a sebaceous. Now these apocrine glands have very limited locations in your body. They are in your armpits, your axilla, in your anal genital area, and surrounding the pigmented part around your nipples, what's called the areola. Okay. And these actually don't develop until puberty. Okay. Now these have a different type of secretion than the eccrine glands. And so their sweat actually gets metabolized by bacteria. And that's what gives that little musty odor. That's the nicest way I can think of saying BO, okay? And so this explains why when kids are running around and exercising and when they're 8 years old and they're super sweaty and they're working really hard, they don't stink at all. And then by the time you're 13 and you do half the amount of activity, you can really reek of BOs because your body has then developed its apocrine sudoriferous glands. All right. Now, in addition to exercise stimulating these, stress stimulates them, pain stimulates them, and sexual arousal stimulates them, which is why some people really like the muskiness of their partner, and you can smell them on the pillow or whatever. But anyway, I have not even going to talk about that anymore. Okay, so with that, we are done with the integumentary system. So I want to thank you very hard for continuing. Thank you very much for continuing to work hard in this new online component to the class. And I will see everybody in class on Wednesday. Do not forget to watch the lab PowerPoint videos on the integumentary system and to do APR number 3 on the integumentary system.